name is Rodrigo Almeida. I'm a professor in the Department of Environmental Sciences at UC Berkeley. There has been an increase in the incidence of Pierce's disease in the North Coast, both Napa and Sonoma valleys, and our project is aimed at trying to understand why or what are the factors driving this increase in disease prevalence. So the project has a few different components to it, but one of the main components is that we hope to perform a lot of field surveys for both insect vectors and the riparian vegetation associated with vineyards with a lot of Pierce's disease, in addition to mapping disease vines in vineyards. And the ultimate goal is to try to correlate all these different factors and see if we can identify the major drivers. The work is very relevant right now because we've seen this jump in the amount of disease in Napa and Sonoma, and unfortunately we don't really understand why or what were the factors driving this increase in the number of diseased vines. So the goal of the project is to try to understand why the number of plants sick has increased so much, and by understanding why, we might be able to develop management practices that are successful in controlling the disease. The project is looking at the traditional insect vectors of Xylella in the North Coast, primarily focusing on the blue-green sharpshooter. We're also looking at other vectors associated with grasses that are present here, like the green sharpshooter and the red-headed sharpshooter. We've also started to do some work with spittlebug vectors, which is a group of insects that's poorly understood, especially in relation to transmission of Xylella fastidiosa. And that's work that is currently ongoing to see if those insects, which are vectors, if they actually have an epidemiological role. In addition to that, we hope to be able to look at some other questions associated with this specific outbreak, such as genetic variability of the pathogen in the region, and maybe determine if there is anything that's fundamentally different about the pathogen itself, or if this is just an outbreak associated with ecological factors such as colder or warmer climates, or potentially more vectors and other components of disease ecology like those.